Hello, and welcome to the next video in the MiGA tutorial series. In this video, we will discuss how to evaluate the quality of your assembled genome sequence. For this analysis, we will head to the same results screen, which was used to infer taxonomic classification. Quality of query genomic sequence, or the genomes in the reference database of MiGA, is calculated by a simple statistic which relates completeness and contamination. Completeness is measured by the presence of 111 single-copy genes, which are observed across almost all prokaryotic genomes, while contamination is measured by frequency that these genes are present in more than one copy. Our example genome is a high-quality assembly, as it contains 106 out of 111 single-copy genes. It also shows redundancy of 3 of these genes, with each one showing 2 copies. Quality scores are then calculated as completeness percentage minus 5 times contamination percentage. This frequency of duplicated genes can also be referred to as chimerism, as it usually, but not necessarily always, is indicative of a chimeric combination between the sequence of the main genome and other highly similar but distinct sequences from other organisms. Chimerism can arise during binning or can reflect actual DNA contamination depending on the source. It is important to note that this is a universal attempt at quality assessment. Further analysis via techniques such as single-cell amplified genomes can confirm if the multiple copies of traditional single-copy genes are real or the product of chimerism. This chimerism can also be explored further via the MiTaxaScan tool. MiTaxaScan basically scans the whole input genome in windows of 10 genes and determines the likely phylogenetic origin of each window. Subsequently, MiTaxa then flags windows that deviate significantly from the genome average. Thus, MiTaxaScan is a CPU-intensive task and an optional analysis that a user has to request by checking the corresponding box. When the analysis is finished, a PDF report will be generated which provides information on the distance of each window from the average of the genome and options to check the origin of each window, download the corresponding genes, or other options. Windows that deviate from the genome average may represent cases of recent horizontal gene transfer or contamination. For further details on MiTaxaScan, the readers are directed to the information online in MiGA, as well as the original MiTaxa publication. Finally, information on gene prediction and assembly length of the input genome sequence, including an option to download all this information, is provided below. By utilizing the available information generated by MiGA, users can thoroughly evaluate the quality of their assembled genomic sequences and even make further inferences about the success of different assembly or binning pipelines, for instance. In the end, areas of chimerism should always be manually examined by the end user and might not always indicate diminished genome quality, but cases of recent horizontal gene transfer or other naturally occurring genetic deviation.